Well, no transcripts working. Thank you very much. Recording. Hello, everyone. All right, so thank you very much. And of course, traditionally, we start off with in recognition of Aboriginal and Torres Strait Islanders people's spiritual connection to country and in continuing ACE's commitment to reconciliation, it is customary to acknowledge country as we pass through. Today, we acknowledge and pay our respects to the First Peoples, to traditional custodians of the lands and waterways. We thank them for the continued custodianship. We acknowledge and celebrate the continuation of a living culture that has a unique role in this region. And we are on Camaragal country today. We also acknowledge elders past and present and thank them for their wisdom and guidance as we walk in their footsteps. So before I hand over to Tom, welcome everybody today for this Brown Bag Open Mic session. Please help yourself to sushi and food as we are here. And without any further ado, I'll hand over to Tom.
Hey, everyone. So today I'm going to, walk you through what I did before I joined the wonderful place that is IPPE, and go through some of the stuff that I did for my PhD. Which is all about spiders. So apologies in advance to any arachnophobes for the number of pictures in the talk like this. Bit of
Right. Okay. Yep.
Okay. Bit of an overview of what I'm going to talk about today. I'm going to go some of the background to my research and what led me to research spiders for my PhD. And then sort of a summary of each of the studies that I did, an overall outline and what the end result of it was. And this is for Darling. This is one of my favorite species of spider, which flows down. These are called ogre-faced spiders because they perpetually grumpy. So this is basically the PhD student as a spider. So from the research background, I did my PhD at Macquarie University between 2018 and 2021 under Professor John Alroy, who you probably don't know. So his research was mainly focused on mass extinctions and species diversity and sort of macroecology, big data in biology. So this is an example of one of his most famous papers. It was from 2008, where he basically looked at the number of genera that existed in the fossil record over the past 500 million years. So it's sort of a map where you can see this is, where mass extinctions occur, and you can actually see this in the fossil record because the number of things that existed in the fossil record dropped really quickly. So I kind of had a similar interest in sort of big data in the aspects of biology, and how we could apply that to things specifically in some contexts with climate change. We can look at what's going to happen with all the stuff we're doing to the world. So why spiders in particular? What drew me to them as a group? You can see from the top image here, spiders basically found everywhere other than the poles. Whether it's, I know Australia in particular is quite famous for all our spiders, but spiders can be found pretty much everywhere else on the planet. There's about 53,000 known species of spiders. Like with a lot of groups, that estimate is seen to have been probably generously half of what really exists to maybe, a tenth of what really exists in terms of the number of species. And for example, for comparison, I think mammals, which is a taxonomic step above spiders, there's not even 10,000 species of mammals that we know. As far as we know, there's only one mostly herbivorous spider, Bagheera kiplingi. It's named for the guy who wrote the Jungle Book, Rudyard Kipling, and it mostly feeds on sap. So 1 out of 53,000 is herbivorous, so they're pretty much all carnivorous, but they can look really, really different. Why, when they all kind of fill a very similar role ecologically, can they look quite different to each other? That's the bonus point for me picking this was I wasn't arachnophobic. Still marginally arachnophobic, but it doesn't quite fill me with the same visceral fear anymore. So getting into the actual outline of the thesis, I had, introduction and synthesis chapters either side, but I did a total of 5 studies through my thesis. First study, we were looking at species richness, so diversity patterns in spiders. We focused pretty much all of the research on Eastern Australia because we were too poor to go anywhere else. And then once we know sort of where the spiders are in East Australia, what's their diversity patterns, we can start to look at some of the drivers behind their body plan and their body structure. And to start that, we looked at intraspecific variation. So if you look at a specific spider species, how variable is their body plan, so that we could say, oh, individuals are representative, which was helpful. So I didn't have to measure multiple in these following studies where we look at a lot of spider species across the whole of Eastern Australia to see if there's any spatial or environmental trends. So do we find spiders of a particular shape in the tropics? And then looking at whether there's any effect of their evolution relationships or their hunting methods. So where builders built one way, burrowers built another thing. And then deep dive into the sort of modularity, or how do those various body parts develop? Are they independent of one another? And if you make a change in your body size, does that mean you'll also have to make a change in the leg size? So what we wanted to get out of this is, because morphology and ecology are very tightly linked together in biology, what you do, so you, for example, if you look at a fox that's in the desert and a fox that's in the Arctic, they are basically the same animal, but because they live in 2 very different environments, their body structure is different. The desert fox has big ears and longer legs. The Arctic fox has very stubby ears, more round body. So that's one thing. So, are spiders similar in that regard? Do you find these ecological signals? And then also, because an ambush hunter will build a certain way to one that actually runs after its prey. What are the differences that we find there? So we wanted to investigate those ecological and evolutionary controls on spider diversity and their morphology and body structure. And to do that, we explored some of the things I talked about on the previous slide: the intraspecific variation, the environmental influence that might have, and sort of their functional anatomy, ratios between body parts. So some key concepts and methods in this that you guys might not be familiar with because they're biological ones. We have the latitudinal diversity gradient, which is the general consensus that there are more species, or tropical areas are more diverse than Arctic areas. And multiple groups follow this trend, and we were expecting going into this that spiders would also follow this trend, but do they have any other trends as well? Because some groups do have nuances to trends. It's not just straight, tropical to Arctic trends. Then we have morphospaces and morphometrics. So this is a form of multidimensional scaling where you represent your data on a 2D plane, and each data point is the coordinate of that individual in that 2D plane. And they get those coordinates by morphometrics, where you will take a bunch of measurements from, in this case, a spider, and combine those, and depending on how different they are to the measurements of every other spider in your data set, that gives you the coordinates in n-dimensional space. We only ever did it up to, I think, 4 axes, because after that point, the amount of variance that gets explained is very, very small. And then the sort of the role of phylogeny or evolutionary relatedness, how we classify things, and hunting strategies, how that goes into things there. And as I've mentioned, a lot of this is under the umbrella of climate change and biodiversity loss, because gets talked about a lot that we're going to lose species in climate change, but there's multiple kinds of diversity, because there's actual biological diversity, number of species, and there's things that we were looking more at, like functional diversity and morphological diversity. So what strategies or what life modes are we going to lose as a result of climate change if we find only certain life strategies are only used in wetter environments or colder environments. So on to an actual study. The first one where we looked at species richness and environmental drivers. So we wanted to assess the sort of species richness gradients, environmental gradients for spiders across Eastern Australia. We focused in on Queensland, New South Wales and Victoria. I think technically the ACT gets encompassed by this too, but that's where our main focus was. We used an online record database called the Atlas of Living Australia, which is a big repository where a lot of institutions like Australian Museum, but also independent scientists can upload records for biological specimens they've captured or find, including where they found it, species identification, sex, etc. Very rarely would they have actual measurements or anything like that in there. So that gave us all the 3 states we looked at, something close to 120,000 records across 83 biological families and over 2,000 spider species just within that area that we looked at. So we then what we did once we had that, we divided our sort of study area into a grid of quarter-degree latitude, longitude cells, and that gives us about 7,400 of those quarter-degree cells that we took species richness from. So for each cell, we'd say, okay, according to the records database, how many different unique species are in that cell? And then a common biology thing is the number of species you actually observe in a location is not the number of species that are really there. You think you're getting less than really there. So we used a bunch of richness estimators, which take an observed richness value, plug it through a formula to give an estimate of what you might think the richness is. And then we use those observed richness and the richness estimators against a bunch of environmental variables that we turned into factor scores and compare them to richness, because a lot of the environmental variables are collinear. So we ran it through a factor analysis because that reduces the impact, or eliminates the impact of collinearity on what we did. So in terms of the richness estimates, so on the left here, we've got observed richness. So this is the actual amount that was in each cell. And then we've got 4 of the 5 estimators here. The 5th one's quite similar to this one. So I just omitted it for brevity. And you can see that it's quite patchy, even from the observed richness. And a lot of these will knock out very low richness cells by design. It's like, well, you only observed 9 species there. That's probably not a legitimate sample anyway. And it kind of the formula will knock it out or give it an error when you have something that's that low in terms of observed richness. So you might be able to see, particularly on this one, 3 red dots of quite high richness. Now, if you know a map of Australia very well, you'll know that corresponds pretty well to Cairns, Brisbane and Sydney, which tells us scientists are lazy. And that basically a lot of the sampling effort gets concentrated around high population areas. And no one wants to go into the middle of the red centre to sample spiders. So some of this some of that impact gets eliminated by the richness estimators, but there's still an element of we need to sample better at the interior. There's a very nice coastal band that we can use, but there's a bit of an issue there with the interior of Australia. And then, so with the factor analysis, so this is just the loadings once we turn the environmental variables into factors. And we kind of emerged there were 3 main factors. The first factor is our classic tropical factor, in that we've got temperature, latitude, having high seasonality of precipitation. So you've got a solid wet, dry season. So that's kind of the behavior we expect from tropical areas. Second, we have our rainy city factor because population density has a bit of an influence here, but it's also where you get a big influence of precipitation, and you've got a big, strong negative influence of temperature seasonality. So you don't want this big sort of difference between sort of summer and winter in this factor. And then the last factor is sort of our alpine factor, in that population density goes down in mountainous regions and vegetation. So that's our main sort of three factors. And then we did linear regression against those using the richness estimators. And what we can see is that they're quite strong positive relationships with both the tropical factor and the rainy city factor when you look at the richness, so species richness. So as sort of precipitation goes up, and a lot of those factors around that go up, then richness goes up. Similarly, as you get into higher latitudes and higher temperatures, you get higher richness. Now, the alpine factor, none of these are significant. There's very, very small negative trends, but none of them are deemed to be significant. So as far as we know, there's no real influence of the third factor, but if there's some of them are being on the borderline of significant negative, which kind of makes sense because you wouldn't expect as much diversity in a high altitude environment as compared to a lowland. So what are the sort of main implications out of that? You definitely want to use multiple estimators when estimating species richness, because each of them has kind of their own flaw or foible, and it's a thing that my supervisor had been wrestling with, trying to find the perfect richness estimator equation for the past 25 years. Because some of the estimators we used in our analysis were invented in the 40s and 70s, and they're still regularly used today in diversity literature. So they're still considered to be really robust despite their issues, and no one's really come up with a better solution. Spiders kind of have a more complex biodiversity gradient than generally accepted because they have the classic latitudinal one, but they also have this precipitation one that's not always considered to be a thing that happens. And that might be a result of their biology. There's a lot of those sorts of organisms that are quite vulnerable to things like desiccation. So it makes sense for them to not be as common or as diverse in a desert environment. But tropical and wet environments tend to have more sort of structural diversity in them, or sort of there's more kinds of plants, there's more kinds of prey. So that it makes sense that a predator group like spiders would be more diverse in those sorts of areas. And because we know there's some environmental signals, we can maybe say, some climate change will result in some biodiversity changes, because one of the big things that is coming up with a lot of climate change is increasing aridity of places, dry seasons. So that was study 1. On to study 2, where we looked at intraspecific variation. So this is, I just had, so it's about assessing how much individuals within a species are varying in terms of their body plan. We just had some widespread species. We can see from here where they were located, the different colours and dots there. So we only used 8 species in this analysis, but we had 145 individuals. So it's 15 to 20 individuals per species. And then we conducted this morphometric analysis of body and leg measurements. So we measured, length and width of the 2 major body segments, and then the length of each of the 5 major leg segments. So that's femur, patella, tibia, metatarsus, and tarsus, for 1 in each leg pair. So that gives us a total of 25 measurements per spider. That then we feed it into the morphometric analysis, and it gives their coordinates. So a lot of time we would do what's called a principal components analysis for this kind of thing, which you may have encountered before. However, principal components analysis doesn't like missing data, which when these are museum specimens that are missing legs, sometimes because they've been in the collection for 80 years, you have to do something that can account for missing data, and that's why we use the principal coordinates analysis and said, I didn't break any specimens myself. It's just, I mean, I think the oldest specimen I measured was 120 years old. It had been in the Australian Museum's collection. So over time they'll lose. And sometimes someone's doing a specific thing that will require them to take a leg off and measure it separately or do something with it separately. So we had to rely on something that could account for missing data. So each of these ellipses is a 95% confidence ellipse around each species. So you can see that each of our 8 species separate relatively well within the first 2 axes of the morphospace. Now, this first axis, we consistently found this was their relative size to each other. So in all of our analyses, we did a secondary data set where we standardized size, which is basically, okay, give me the body length or one particular measurement of one spider in the analysis and change every other spider's measurement for that particular body part to that number, but keep the ratios between all its other body parts the same. So they kind of adjusted them to all be relatively the same size, but they keep the ratio between all their other body parts. And this, as you imagine, because we've taken away the thing that most accounts for a lot of the variance, it does cause more overlap. But they're still quite distinct from each other. So colors mean they're part of the same family. So these are orb weavers in the blue, and I believe they're wolf spiders in the black. So you can see that sometimes it's getting size adjusting makes some of the families overlap more, which is interesting. And then on the right, we did a linear discriminant analysis, which basically said, okay, here's a particular spider, tell me what species you think it is. And this was the probability of it getting it right for each species. So there's a couple of species that are not as good, and they're usually, and I think it's these two are overlap pretty much entirely here. So that kind of makes sense. Oh, that's why those two they're either fulfilling a similar niche, or they've got very, very similar body plan. So that was kind of the main, but we thought from that individuals fairly well represent their species morphology fairly well and consistently across geography. That was the reason why we did it over such a big space. It was just in case there was some geographic signal. So that helped us to or support the idea that we could use individuals representatively in analyses going forward. And here's another example of one that's missing a leg. So I think that's an orb weaver or something with widow. And so that's kind of what I would have to deal with sometimes and try and get the measurements. So that it helped us a lot in this study too. Was kind of like a confirmation. So it's like, oh, can our approach that we want to use for the rest of the PhD work? So when we got into study 3, this is the real meat of the of what we wanted to do. Was so we had some study 3 specifically looking at variation across space and environment. So in between that north-south from tip of Queensland, bottom of Victoria, is there a difference? Do we find similar patterns in the environmental variables morphologically as we did with diversity? So we had a total of 553 spider species in this part of the analysis, each with those 25 measurements. Most of these I measured through photographs digitally. Some I had measured through the microscope with, there's a ruler you can get on a microscope eyepiece that as long as you know the scale of that, you can measure them. So it involved, a lot of photos, and some were from literature, specific species. You could get measurements out of the literature too. And so here's a bunch of the kind of photos I would take, and even now you can see the difference between what spiders can look like. This is a huntsman of some description. That's an orb weaver. Not 100% sure what that one is, but might be an ant mimic or something like that. So yeah, we use the same ordination techniques that we did in study 2, along with spatial autoregression. So when we were comparing our designated sites that we made at the eastern coast, we did this form of spatial autoregression that accounts for the two sites being next to each other. So if two sites that are next to each other are really different, it will weight that more or less depending on their distances. So but the main thing that we found was nothing. There is no environmental signal, and this is just to show you all the non-significant p-values that we had. Annoyingly, that one's very close. So yeah, we didn't really find any sort of pattern between morphology and environmental factors. This was true when we did it using the entire data set versus we singled out particular families, like we would just do it on the jumping spiders, and we still didn't find anything. There's some very minor differences between sort of leg structure, when you look at leg structure, and range size. So spiders that have sort of longer functional components of their legs compared to the structural components tend to have wider ranges, and that the spiders that fall under that are things like orb weavers. So there's but it's very weak, and it's only in one area. So we were kind of like, we're not even sure this is just because we did so many comparisons that one of these turned out to be significant. So kind of like our main this is there's no strong environmental signatures in spider shape or size. Nor do we have a trend of site species composition. There wasn't a massive sort of turnover where we'd suddenly stop finding a bunch of species and then find a completely different set of species at a breaking point, which you find in some groups. So it definitely suggests that the body plan is way more influenced by evolutionary constraints and ecology than sort of this environmental. And here's the sidelong view of that spider from the start, the ogre-faced spider. So they look quite different to what, in some ways, you'd expect a sort of archetypal spider to look like. So on to study 4, where we looked at the effects of phylogeny and hunting method on the body plan. So this is a fairly rough phylogeny of spiders that we used to, similar to one we used to break up. So we used the exact same data set from study 3. We just broke it up in different ways we didn't care where the spiders were from, because we found, okay, that doesn't make any difference. So we broke it up into 7 phylogenetic groups. What the groups are is not super important, other than there's a big split in spider phylogeny between megalomorphs and araneomorphs, and that happens about here. Megalomorphs are the more ancestral ones. So this is the group to which tarantulas, funnel webs, those sorts of spiders belong. And then araneomorphs are most of the other ones that you think of, like huntsmen and orb weavers and redbacks are all in this quite big group. So we couldn't just have 2 big groups like that. That was probably not meaningful enough. So we broke some of this group down into other smaller groups, getting steadily more evolutionarily recent. So some of these groups had only really emerged 4 million years ago, as opposed to megalomorphs, which emerged over 200 million years ago. Um, so in terms of the phylogenetic results, we had a pretty clear infra-order separation. So that's between the megalomorphs and the araneomorphs. So all the megalomorphs basically come into this part of the morphospace. So we have, here relative size, and the second axis is the ratio between body and leg length. So up here, you're big, and you've got a pretty even ratio between your body and your leg size. So down here is where you have a very, very long legs compared to your body. Because down in the bottom here, making life difficult, are the daddy long leg spiders. Because they just, distort things quite a bit. But that's where they sit. So, because you imagine, what that long legs looks like, it's got a very tiny body and these massive legs. So the sort of other groups kind of would separate out as well. So this is the Araneoidea, which is where most of the orb weavers and similar spiders to that fall. And then this is one of the other clades, which includes things. I think this one includes wolf spiders and like that. And then this is the most derived one, which I believe contains huntsmen and jumping spiders. So some of them are not as well defined in terms of there's a lot of overlap, but there is a pretty big sort of separation between the megalomorphs and the araneomorphs. But of course, that's only half the picture, because we also looked at hunting methods. And this is where we divided them into a total of 9 hunting methods. Things like burrowing spiders, whether they're using, an orb web like St. Andrew's Cross, or more of an irregular web like a redback spider that you find in a corner. There's not a lot of organized structure to something like that. Whether they just hunt along the ground, like a wolf spider or a huntsman. They don't use webs in prey capture. They pretty much only use webs for egg sacs. Or if there's some form of silk use, but not necessarily for a web. So there's a couple that are like the poke-faced spiders. They're net casters. Is their more official name. And that they actually build a little web net between their legs and fling it at their prey to catch them. So they're an example of something that uses silk for prey, but it's not strictly in a web in the traditional sense. So again, we kind of have this split between sort of non-web builders and web builders. You'll notice that a lot of the ones that are like burrowing and retreat spiders are also megalomorphs. So that tells us that conditional, that way of hunting is more ancestral, which correlates with what we know, because it's generally accepted in spider evolution that silk came first for other reasons beyond prey capture. It was probably evolved originally for looking after the egg sac, and then they're like, oh, this is quite sticky. Eventually spiders evolved to use it for prey capture. What tends to happen is non-web builders have the more balanced body plan of having an even ratio between their body size and their leg length. And there's some hunting methods that are pretty unique to certain phylogenetic groups, like ambushing with silk. There's one family, Sidaridae, that will be a lot of the ones that we've got. One hunting method here, mimicry. They're pretty much an entire family of ant mimics. So their body structure makes them try and look like an ant, so they can go into an ant hill and take out their prey. And some of them have evolved the behavior that they will, once they've actually killed their prey, they'll sort of carry it out of the nest. Which is a common behavior for ants generally, to carry deceased members out of the nest. So they're doing that. The ants are like, oh yeah, you're just doing the right thing. But meanwhile, it's just killed that ant. That's why it needs to take it out of the ant nest. So that is what we've kind of got some instruments. There's both an effective phylogeny and an effective hunting method. There's you've got a lot of similar body plans that will cluster together both within those hunting methods and within families of particular spider groups. So this suggests a strong effect of functional convergence, like you will develop evolutionarily the same tools for the same job. So if you are an ambush spider, even though you might not be related to another group of ambush spiders, you're likely to develop similar body structure because there's a sort of ideal body structure for sitting in the middle of a web or being an ambush hunter. So here we've got an example of a crab spider where they have these very big, robust front legs because they're an ambush hunter and they sort of pincer grab their prey. They also have a lot of camouflage stuff as well. So this suggests, I would suggest kind of like that there's this evolutionary constraints and sort of also play a key role in that as I said, the megalomorphs kind of occupy their own space and there's not a lot of non-megalomorphs that enter that particular area. So there's certain body structures that are linked to parts of the spider family tree, and there's certain that are linked to their hunting method. So onto my final study, we looked at the modularity. So are the various body parts of the spider independently variable? So if you have your front legs getting longer as part of evolutionary pressures, are there similar changes in other parts of your body that seem to go along with this? So we flipped the data set from study 3 and 4 to 25 measurements rather than 553 species. So before every data point was a spider, now every data point is one particular body part measurement. We then added a second data set from a book about riceland spiders in Southeast Asia that had a total of 211 spiders with measurements that we could add to this. And then we created morphospaces again using the body parts as separation rather than individuals. So here we have the various body parts. So we've got a very clear sort of body module here. Like these are all the body part measurements. They kind of sit by themselves. And then we have the leg measurements sort of being in this arc along here. But at the top here, we have most of the structural elements like their femur, tibia, and metatarsus, which are what give the leg most of its length and structure. They don't provide much of a functional purpose, which is what the other two measurements are, the tarsus and the patella. So that's, determines how the spider's leg can bend, how good it is at grabbing things. So those we termed the functional leg components. And then we did a bunch of test spaces to say, okay, treat these as groups. Are they statistically different from each other? Using both this standard one and one where we did the size adjusted data again. So in pretty much all cases, they are distinct modules. Occasionally, they're serving on the first axis. They're all distinct from each other. Sometimes on the second axis, they're not. And this is usually when we include the size adjusted data. And because as I've said before, a lot of the variance gets explained by spider's body size, because you can have a big size range in spiders. So this told us that, okay, these particular modules would vary independently of each other. So just because your body size is getting bigger doesn't necessarily mean that there will be an evolutionary pressure for you to also develop longer legs. So that kind of tells it. And what parts of your leg would change according to evolutionary pressures would depend on whether, is this something that might influence the structural components, or is this something that might influence the functional components? So yeah, this shows we've got sort of very clear modules, and there's a separation of functional and structural components supporting ecological adaptation. because the functional components, for example, if you trying to develop, or you've got an evolutionary pressure to develop a certain hunting method that might require certain functional changes, that's a bit more of a pressure on that. And this is consistent across geography and families. Again, we tested things using just particular groups. So if we did the just using spiders that were orb weavers, just using spiders that were jumping spiders, and we got the same modules regardless of whether we're using the whole data set or those particular publishing groups. And then sort of to wrap it all up science-wise, spider diversity itself shaped by multiple interacting factors, what we found. Body plans are driven by both evolutionary constraints and hunting methods rather than environment. Climate may impact future biodiversity and morphological diversity patterns. The biodiversity is pretty much a given because we've seen a lot of this already in terms of range changes, particularly in other parts we've seen mosquitoes' ranges expanding because temperatures are warmer. And the morphological one we're less certain on, but we think there might be some chances. So overall, this has given a bit more of a clearer picture on how spider functional evolution and macroecology works in Australia. But of course, it's just Australia. So there's still some unanswered questions around this. And the main thing is, do we have a true arid gradient? Because as I told you earlier, scientists can be lazy. So if we had a bit more of a sampling of the arid interior, would we have a different picture? Is this true for other regions? So we did look at WA. Would it have a similar pattern? You know, would so if you looked at the same thing along the coast of, say, South America, would that have a similar pattern in terms of the diversity? And is this true for other arthropods? So if you looked at beetles, would they have a similar sort of divide, both in terms of their diversity, but also in terms of their functional aspects? So would we find similar body plan modularity in beetles? So those are some of the things that, yeah, we answered some questions, but of course, you answer one question in science and it generates 4 more. So that is all from me. So if you guys have any questions, I would be happy to field them.
A whole round of applause for Tom. Thank you. Interesting. Questions from the audience?
Of course you do have.
I'm curious, is there a relationship between size and poisonous?
Oh, generally, yeah. So in terms of how much venom they have, it's sometimes yes, sometimes no is the thing. So we think of, if you think of 2 quite big groups of spiders, tarantulas, which are generally regarded to have not very severe venom, and then funnel webs, which are regarded to have some of the most lethal venom in the world, and they're quite similar sizes. So what tends to be the case is if you're very big, you probably don't need super powerful venom to take out your prey because you're that big. If you use a web, you tend to not have as powerful venom. So even the biggest orb weavers don't have particularly powerful venom because the web is doing a lot of the subduing for them. And then so whereas, you know, funnel webs are a bit of an outlier in that regard, they're quite bad venom-wise, but then again, they're only really bad for primates. There's certain venoms that are quite bad for primates, whereas, you know, if your dog if a big dog gets bitten by a funnel web, it's probably not going to die. It's going to be sick, but it's not going to die. Whereas we get bitten, we die. So, so yeah, it's complicated. It's much more tightly linked to where they are in the evolutionary tree than their hunting method than their size. They're particularly fair.
Thanks.
No questions?
Yes.
Can you tell a gender. Can you tell spiders by their gender now? Are you like an expert at gender? Gender?
So you can, in terms of, so I probably said we used, in this analysis, we only used female spiders.
Oh.
So we only used adult females. You can tell an adult female from an adult male because there's a particular body structure which we didn't measure called pedipalps that are much, much bigger in adult males. But you can't tell the difference between a juvenile male and a juvenile female because that body structure only really gets that big as the male gets to adulthood. So as it goes through them, it's adult molting. So yes, you can tell the gender of the sex of a spider. It's quite difficult. So that's why we just kind of like, let's just stick to one particular. And in all cases, someone else has identified it as being that sex already. I didn't do it myself. Mona, and then Karen had a question. Yeah. Oh, they're
Solitary.
yeah, they're generally very solitary. You'll get because they tend to have lots of eggs in an egg sac. When they're very juvenile, they're a bit more communal. And there's certain species that are less territorial than others. So you might notice, on the brickwork of a house in Australia, you find lots of black house spiders and things like that. They're probably quite related to each other. They don't tend to disperse far. But then there's a lot of species that are quite antagonistic because in nature, any other individual, especially of the same species as you, is a potential threat to your food resources. And this is what's fueled that reputation amongst spiders for sexual cannibalism and things like that, that they eat their mates after sex. And this is really only observed in like a couple of species. But it's mainly that the female spider sees the male spider as a threat to her territory more than anything. But then there's other species that are so solitary. There's a species of cave spider in Tasmania that it can't afford to be territorial because it might only encounter another individual of the same sex once in its lifetime because it lives way back in a cave. So yeah, generally they're pretty solitary. But there's a handful that are slightly less solitary than others. Karen? Yes.
Yeah, I've got two questions.
Yeah.
So you said at the end there that the hunting method and characteristics of the spider were more related than environmental factors. To what extent are those two things related to each other? Do spiders in northern areas have more activity than spiders, or just
So there'd be a small relationship in that you'd probably find a greater variety of hunting methods in a tropical environment, and that's because in a tropical rainforest, you've got more variation in the sort of surface structure. You've got, the leaf litter, understory canopy kind of thing, versus a temperate forest where you don't have that as much. So you might find a greater variety of hunting methods in a tropical or warmer environment versus a temperate environment. So that would be a small relation, but in terms of, our analysis, we didn't necessarily sort of see, okay, is there a tight link between these two?
And my second question is, if you could scale a spider up to the size of a shark, and a shark down to the size of a spider, which one would win in a fight?
Well, okay, assuming a hypothetical world where a spider can be that big, because it has an exoskeleton. So if you made a spider that big, it would crush itself. It also couldn't breathe because it doesn't have true lungs. So assuming it could survive at the size of a shark, the shark would probably win. The shark is way more mobile. And the the spider, like, so, the megalomorphs especially, the like polymerphs, they only have really one attack method, and that's to rear up and punch down. Their fangs point down like that, and that's the only thing they can do to attack. Whereas a shark can do a lot more than that. The other group, the uranomorphs, their fangs move like this, so they're more of a pincer movement, and that's where you get the huntsman and that are probably closer to in ecological niche to a shark would be like a huntsman or a wolf spider that runs around and just jumps on its prey and takes it out. But even then, I think that because you break one leg off and the spider's pretty much done.
What do you think they don't have regular lungs for the spear?
So they don't actually have lungs like we have lungs. They have this thing called book lungs, which is kind of like a moist underpatch on the underside that sort of passively absorbs oxygen. So they don't have active breathing. So they're a step above most insects, which actually just have spiracles, which basically holes in their exoskeleton for oxygen to passively pass into. So they're a little bit more active breathing than that. But yeah, they can't actually breathe in and out actively the way we can. So, or the way a shark could. So that's the spider would feel puffed out very quickly at that size, but it just couldn't get enough oxygen into people's cells.
What is vision? Do they have great vision?
Some of them do. So the one I showed at the start, the overface spider, they've got, because they're plants at night, they've got fantastic vision in those two big eyes. They get rid of their retina in the morning because it's too energetically taxing to have that during the day when they're not using it. So yeah, some species of spider have pretty good vision for it, but they still have compact eyes, so they're still not going to be as good as at distance perception and image resolution as a vertebrate. Yeah, Aaron?
I'm just curious about your some initial motivation to study
Yeah.
why you chose spider,
Well-
especially.
Well, so I do kind of say they're found everywhere, so they're a great kind of group to look at for this at this big scale. Because if you can find something everywhere, that either means they don't care about where they live, or that you find all these sections about they're very good at adapting to where they live. And we found that they generally don't care that much about environment. So that's kind of part of the motivation. And yeah, my supervisor would have been happy with me picking any group. He personally studies mainly mammals and plants.
Yeah.
So that was his area. And yeah, so I was just picking a group. And yeah, the arachnophobia played into it a bit. It's like, you know what? If this doesn't cure me of that one. Jesse?
If I want to write ARC research grant on spider,
Yeah.
how can make it as a successful application?
So a lot of what we were talking about is spiders are really important as pest control. Like they're a very fantastic biological pest control that's not bad for the environment. And you know, we have, especially in areas like Queensland, where we have big farming issues with fruit flies, things that, you know, introducing spiders into the mix is great. There's also lots of medical applications to their venom. So anything we can do to understand them more can help in those scenarios. Also, there's the classic thing about spider web being fantastically strong, having really good tensile strength. So there's a lot of studies going into that and how we can replicate that structure, and you know, one of the pluses and minuses to it.
So on that note, we've got other meetings coming up.
Yeah.
So I'd just like to thank Tom in a minute, but just to let you guys know, in July, we've got a few international guest speakers coming from overseas to do that. And then late July, August, September, we've got all our wonderful PhD students that are here and online doing their mid-candidature reviews. And then in October, I'll be doing the other half of the vertebrates. So I'll be doing the other bits. Yes. What Tam was talking about. So I'll be doing what I did for my PhD in October.
Your first PhD.
My first PhD. Well, round of applause. Thanks so very much, Tom. Thank you, everybody online. Thank you.
Are humans the main predators?